from infected menhaden, because you can find a lot of different strains of the bacteria in menhaden as well. So that's another pathogen. So as far as when you looked at, you go, I had the submissions from the DEC and I have another submission from colleagues in Rhode Island, but when you actually look at, in Google, menhaden kills over the last year, they actually started in the spring and there were huge die-offs down off of North Carolina. And then this is one of the first articles they started talking about this swimming in circles thing. This was off New Jersey in the Hudson River. We actually had a citizen that submitted a video to Kathy Hadela in the Hudson River unit and clearly shows the exact same signs. Even in Connecticut, I have colleagues at the University of Connecticut, they were getting submissions from menhaden kills. This is the time, it was like late May, early June. This was where it began in Rhode Island, so fish were collected from there as well. And then sort of the biggest kills in New York were actually in the Peconics. And those were really well documented. Actually, the DEC put out a report last week. If anybody hasn't seen it or would like a copy, just email me and I will send you a copy. It's really well described what the conditions were, the algal blooms that were occurring, the oxygen levels, and even our diagnostic report is in the appendix. So algal blooms probably were to blame for part of this, but as they go on, I'll tell you that it's always a combination of different factors that cause these kills. So I'm just going to show you some pictures. So this was off during the North Carolina outbreak. That's not a wave there. That's all menhaden dead. So this is from New Jersey, Rhode Island. This is off in the Peconic. More pictures from the Peconic. Basically just showing you some dead fish. So we received fish from the Hudson, from near Menhaden and Peconic. So we worked those up, as well as a submission from Roxanne Smolowitz. She's at Roger Williams University, and she sent us some isolate or some samples as well. So I'll get into the different conditions that we saw. Basically, the external observations were ecchymosis or hemorrhaging near the vent, hemorrhaging in the eyes, the skin, the fins. I'll show you pictures. Hyperemia around the nares, the nose of the fish, and this abnormal swimming behavior. Not every fish had that, but each site was reported. So I was going to try to show you some videos of this, but after the difficulties we had with the computer, I'm not even going to try at all, so don't worry. I do want to say that as I was looking basically on the web for people that have been documenting the different menhaden kills, in December, there's a scientist on a YouTube video off of Mobile, Alabama. He's got hundreds and hundreds of menhaden dying around him, smaller Gulf menhaden, and they're showing the same circular presentation that I've discussed already. And he was saying that there was no red tide there, and this was, he'd seen fish dying in red tides, and they just do not behave that way. So it's not an algal thing. So here's some pictures. These are from the DC submissions. So some of the hyperemia I mentioned. Some of this bleeding in the eye. So initially that scared me because that was one of the VHS signs. And so that bothered me a little bit, but they were actually tested for VHS and that was not it. So the hemorrhaging was not really severe in a lot of cases, but when we did the internal observations, then we saw even more hemorrhaging and congestion on the inside of these. So ascites is just the fluid in the abdominal cavity, so that was bloody. I have a picture, I'll show you the pyloric ceci were all hemorrhagic. even when you open the cranium, we had hemorrhaging around the brain. Some of these fish, the intestines were hemorrhaging, stomachs, gonads. So the gills had sort of an excess amount of mucus on them, and that's never a good thing. So sometimes that's related to algal blooms. So as a way for the gills to sort of, they get irritated and actually produce more mucus than normal. Here's what a lot of these look like. So very hemorrhagic inside. This is not normal. So before I get into the virology, we did the standard bacterial culture. We did skin scrapes, gill clips, looking for common parasites. We didn't see any of those. So the only thing that was in common with all of these is we were taking either organ samples, we would homogenize those, filter them, and then put them on cell culture. So we did that for these pooled organs or a separate brain sample. And so we used these to inoculate basically fish cells that you grow in a flask. And what we saw was these cytopathic effects. So this filtered homogeneous would actually be killing the cells, these fish cells, and it did that in 3 of the cell lines that we use. So this blue BF, that's a bluegill fry cell line. The KF1 is a koi fin cell line, and the CHSEs are a Chinook cell line. So we saw this effect in those. We did not see them in the cell line, which we call EPCs, and that's what we use for diagnosing VHF. So it did not show that effect in those lines. And so Kelly, our technician, made this really nice panel. And so the 3 cell lines that we saw positive CPE are on the top, and then you can see the EPCs on the bottom. Those all remain normal. What you'll also see is that we did not see the effect in the brain from these. These were the 010 and the 011. Those were the samples from the Long Island area. And so I have a magnification of this one in the upper right-hand corner. I'll show you that CPE. And so this is that shot here on the right. Well, the paper I mentioned at the beginning from Stevens, that work was done down in the Chesapeake. And there's almost the identical CPE. So then what do we do after that? So the paper from 1980 suggested it was an IPN-like virus, so a virus. And so we have primer sets so we could do PCR. So we ran those and we did not get a positive result on these. And we continued to passage those, and the CPE would occur in every passage. So it's definitely a viral agent. So it's something that we're still working on. We tried to do EM on it, because that would be a really nice diagnostic, at least show that it was the right shape for IPN. We did do a chloroform treatment, and the inoculum survived that. It still caused CPE, so that shows us that it's a virus without an envelope. So IPN still fits that. Our next step is we're trying to sequence that virus. And so we're still working on that, and I'll tell you at the end, maybe I'm getting, I'll do the histopathology, and then I'll tell you what we're going to do in the future here. So we had some pancreatic necrosis. The pancreas tissue didn't look that bad, but what we did see was this congestion of blood in the organs. And that is often a sign of the anoxia. And we definitely had that going on in the Long Island samples. There was no evidence that that was occurring in the Hudson River sample. So we also had some necrosis that was seen in the kidney and the liver, inflammation in the heart. I'm going to show you pictures of all those. And because they're wild fish, we saw a few parasites, but not too many, and we wouldn't associate that with the kills at all. So this is actually a shot of the pancreas in this dead center. I'm not sure I actually have a pointer. So
You do, it's the center button.
thank you. There we go. So there is some necrosis in here. This is actually the normal vacuoles that are in the tissues that in the belly. So that's normal. A little bit vacuolated, so that's a little bit unusual. So this is some of these are anterior kidney and just some necrosis that's going on there. And you shouldn't see this much blood in the kidney. So these sort of orangey cells, those are the blood cells in the fish, and you should not see that much. So that's some hemorrhaging in here. And the liver should be all this sort of light purplish color. This is congestion. This is probably due to the anoxia. So that was characteristic. And spleen, all those spleens supposed to have, it's a storage organ for the blood. So actually should not have this much blood in it. So this was actually congested as well. And then a little bit of hemorrhaging, those inflammatory cells, you normally don't see those in the heart either. And then this is just a slice through the eye of the fish, that fish that had the hemorrhage. It's just showing you that the fish had hemorrhaging in the eye, so that's not too much excitement. So we still think that the viral infection had something to do with the die-offs. Clearly, Steve Hines, people at Stony Brook, and the health people on Long Island, they documented that it was an oxygen problem. There were still predators there jamming these fish up into the river. There were algal blooms going on that could have taken oxygen out of the water, particularly in the early morning, where you get these oxygen crashes from algal blooms. And then the predators were part of the issue. Water temperatures were increasing at that time of the year, and so that plays a role. What we're trying to do is still confirm that what this virus is. So we're still working on that. I've submitted a SK grant, so hopefully I'll find positive news in May from NOAA that they'll fund some future research with that. So I've lined up basically these NIPS guys that collect Menhaden at the dock as they're brought in each day. And so I have arrangements where they could collect fish for me from 4 different places along the coast to see if we can document whether that virus is in all the sort of, I'm not a good fisheries person, groups of fish that moving up and down the coast. So I just wanted to thank Andy and a whole bunch of the DEC employees that were out there collecting the samples for us. Justin Krebs is a consultant, works around the new Tappan Zee Bridge. So his crews were out there looking for possibly injured sturgeon that we look at, but they also supplied samples for us. So, any questions?
Do you have time for 1 or 2 questions?
Yes. We haven't seen any other species dying at the same time. There were a few, I think, either catfish or bullheads in the the Hudson that were reported during those initial kills, but we didn't look at those. So that would be interesting to see whether we could isolate the same infectious agent. IPN actually can be found in lots of different, not only freshwater but marine species. It's been identified in mussels. It can be found in a lot of different places. So it's just, our view is maybe that's just a virus that's in the population and it's always been there, and you have all these additional stressors that are put on the fish. So maybe that's why it was just seen this year or last year. Thank you.
those of you who just walked in, we are running slightly behind in this session. So without further ado, I'll introduce our next speaker, Matt Fuchsia, I'm sorry, will be speaking to us on early mortality syndrome in Steelhead. Matt is coming to us from SUNY Brockport, and
thank you, Fred. As you said, my name is Matt Fuchsia. I'm a current MS student from the College of Brockport, looking at thiamine deficiencies and salmonids, and we're also doing some fatty acid analysis on the side as well.